Shall I just answer a couple of the questions, or?
Sure, you can answer
How
some
do we go?
questions and then we can go into discussing Toast and showing them how to use it.
Cool. Hello. Okay, so Toast it is an app that we've built to make data provided by NASA accessible without using loads of coding. So it's basically designed in order to enable biologists to do genetics and investigate life's genetic response to stresses associated with spaceflight. So I'm really pleased to have the opportunity to share this with you. I've been working on it for a bunch of years and we've just published it. Did you guys get the paper? Yeah, so you've gone over it so you roughly know what it's about. So we could dive in, just play with it for a little bit, or answer a couple of questions and then dive in and play with it.
So we read the Space Lettuce paper and discussed that as a class on Tuesday. They were supposed to read the Toast article for today, but we haven't discussed it yet. So I don't know if you want to provide another overview of that, and then we'll go play with the tool.
Yeah. And so also, I'm glad that you read the plant microbiome article, because that gives me a chance to again promote one of the apps that we've got on the website, and that is Mango. Have I mentioned Mango before? So we haven't published this one yet, but Mango uses the same kind of infrastructure that we use to make the genetics available through Toast, but it makes the microbiome of the space station available. So in that paper where they were discussing that all the different microbes that lived on the lettuce, on the plants on orbit and those on the Earth, well, we've taken that type of genetics data and made it accessible through the apps, so you can download the data and explore it and do other extra comparisons. With the Mango app, it's just the space station microbiome at the moment. We're still processing the plant microbiome from that paper that you read, and the mouse fecal microbiome, and there's a couple of other ones that are coming from our collaborators at the Jet Propulsion Laboratory, but they take their time. They're blaming the virus. Apparently San Francisco's been hit pretty hard. No, it is LA. Anyway. Yeah, so microbiome, that app is there available. Mango is there to play with too, but we'll come back to that. Okay, so the moral of the story, to be a plant researcher, you don't just go into one box. As you go through college, they say, oh, you've got to be a molecular biologist, you've got to be this, you've got to be that. And you try and put yourself in a box as you go through all these different courses, right? It's not the case. You're not just a biologist. You're not just a chemist. You're not just, you're a scientist. And that means you have to be able to do all these different fields. And what I mean by that is, for me to really understand how plants are going to respond to the space-like environment, I have to do genetics. I have to do microbiology. I have to do all kinds of different fields to get the overall picture so I can really understand my model organism. And that leads us on to one of the questions that you guys asked. So is there a similar thing to Toast for rats, mice, and other mammals? And yes, there is, and that is on my website. That is GeneLab Cross Kingdom Species Explorer. And again, it uses the same kind of infrastructure, the same structure that we use in Toast to explore plants, and it goes into the mice and all these other model organisms. So you got to find what is the model organism that you want to study. I mean, you don't have to be a scientist, but if you do, you end up growing that model organism a lot. So that's why I like plants. So I chose plants as my models because I like growing plants. But some people like nurturing animals. And so, hopefully these apps will give you a way to explore the genetics of a model organism that you can practice looking after, growing, and then eventually doing experiments on. The road to experimental biology is paved with dead organisms along the side roads. I've killed so many bloody plants. Oh dear. Good job I'm not a human biologist. But as you grow them, once you're capable of growing them, then you can really start to delve into genetics and test your genetic theories. So I'm trying to arm you with the tools to start to explore the genetics mentally, but ultimately you've got to do the groundwork eventually and become an experimental biologist to disprove or prove your theories right. Anyway, so that's why we're putting this data into your hands, because we want to get you to do that kind of stuff. Okay, so let's play with Toast. I'm going to share my screen, I think, if that's all right. Any questions on that? I'm just kind of rambling along. You might have guessed. Was that a reasonable introduction? Thumbs up. Okay.
I was just gonna say, like, as you're going through Toast, like maybe the students, you guys can pull up the window on your computers too, if you have enough screen room, and try to work with the tools as Richard's discussing them. Do you think that'll work, Richard, or would you rather they do it after you show everything?
Well, yeah, I like to click along with people. So if you have big enough monitors, if you had 2 monitors, that would be the perfect world, because you could put me and my ramblings on one, and then also have it open on yours on the side, so to speak. Yeah, so feel free to follow along. Feel free to just watch. Doesn't or 50-50, swing back and forth, right? Maybe you have pen and paper in your hand as well. I like to take notes and just scribble stuff down. I find that's actually kind of quicker than trying to type it out. There's a mixture of things. So I've got way too many tabs open. Can you see that? Oh my God, I'm sorry. My poor computer. Okay. What am I doing? Okay, so I'm going to search for astrobiology. Biology.botany.wis.edu. So in the olden days, there was this paradigm that we academics had to deal with, and it was publish or perish. And that's not true anymore. We live in a post-genomics era. So in the olden days, in this publish or perish paradigm, you had to design a hypothesis, design an experiment, and test the hypothesis. Nowadays, in the post-genomics era, post-internet era, that we have these huge data sets that just show us the patterns of life. And then we need people to understand and describe those patterns. So now we live in an era where we can share and thrive. By sharing the data in a way which makes other people discover patterns, we can all thrive together. And then generate new hypotheses from these older big data. And that's why as soon as we get spaceflight data, we like to just put it on the website and try and make it accessible. And that's how we got a job with NASA GeneLab. And so GeneLab, I should probably just type in GeneLab, is a government organization. It's part of NASA that's based out of the Ames Institute in Silicon Valley. And it's full of lots of lovely people who like to try and understand how life is responding to spaceflight. We've been an advisor with them since the onset. And essentially what they've been doing is building a data repository to make data analyzable. So they've been collecting all the data to one location. So over the decades, loads of people, hundreds of people, maybe thousands, did experiments in outer space on loads of different organisms. And this information just got scattered across the web, across a bunch of different NASA bases and international bases. And then some people realized that's bad, and maybe we should try and get all this really valuable data together and get it organized so we can really maximize its value, because the next era of spaceflight exploration is critical that we get it right. And so we should maximize the impact of the previous data that exists. And that's why GeneLab was created. And so now we have a list of about 239 studies. It's all in one location, and you can go through and you can access the data. Most of it is in its raw format, so you need to do bioinformatics, like computer programming, to access it. But the stuff that we've been able to access, we have put on our website already processed, so there's no programming required. But there's various points in my apps that will take you back to GeneLab. The reason is they've done a really good job of curating the metadata. So maybe we're interested in the genetic response of mice skin to spaceflight. And what they've gone and done is collect together all the metadata about there. Like there's a description of how the study was done. They've given it a code name so we can quickly search for it, the GLDS accession code. Then they've tried to come up with a description of all the different factors that were present in that study. My job is to now go through all of these and tell NASA where they got it wrong and try and make them upgrade it. It's an uphill battle, but we're getting there. It tells you the type of study that was performed, RNA sequencing, and it's like a microarray. It's my favorite form of analysis. It does transcriptional profiling, so it monitors all the genes at any one moment. Microarrays, they did most of the genes, but they missed a few. That's the old version of this technology. So this is omics technology, post-genomics. So now we have the genome sequenced, we can analyze all the genes. Dun, 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 dun. So it's got all the metadata. Oh my God, there's loads of it. Isn't there loads of it? It's telling us about the RNA quality, like all the intermediate files that were produced, the different ways the animals were raised, the different types of habitats, the old ones and then the new ones, depending on when they went up. It tells you if they went on SpaceX or space shuttles. It's all there and there's so much of it. So where to begin? So I made an app for that. Okay, so we're going to go to GeneLab, back onto the Gilroy Lab Astrobiology website. I'm going to click on GeneLab. I'm going to look at my GeneLab meta-analysis app. Can you see that button? Okay, so I just made a new app for this. So this probably won't work because the best way to find something that went wrong is to do a live demo. Okay, so what is NASA GeneLab? It is a truly global collaboration. You can go back to their website, there's a link there. So this is an app that I've made to explore all of the data. So let's have a quick look at it here. So at the bottom, we can see all the different locations in the world where this data is being downloaded. So that's why it's a truly global collaboration. So what's happened is NASA have given specific codes to 300 studies. They've assigned them types of projects, so some on the ground, some in outer space. They've described the type of molecular analysis that was done. Most of them are transcriptional arrays, like looking at gene expression. They've listed all the different organisms that are doing it. So this is and then they've sent that data out to all around the world. Anyone can go to their website and download it. But don't waste your time doing that because I've already done it for you. Okay, so we can summarize the data. So some of it is spaceflight, some of it's on the ground. Actually, some of it is in parabolic flights. Most of it is microarray, some of it is RNA-seq, but there's proteomics, epigenetic studies, all kinds of different assays are performed. Loads of different model species. Mouse is the major one, but there's some human stuff, and Arabidopsis is the best. So maybe you like mice. So I'm just going to click on that mouse thing. So now we're going to filter down. So yeah, lots of people in America like looking at mice. Okay. And there's lots of ways of looking at it. The RNA-seq is the new stuff. The microarray is the more traditional data sets. And maybe you're only interested in the spaceflight stuff. So there's like 24 studies that use microarrays to look at mice. Okay, that's kind of cool. Let's have another look. Actually, can you see I'm confined? I've got these like areas on the side here. Okay, cool. So all of these apps, they pop out into another tab like that. Go a bit wider. To improve your user experience, recommend popping out. Okay, so I've got a video. I'm going to play this because I've never had an audience before, and I've always wanted to play this video to an audience. Can you hear that? Oops, my bad. Sorry, one more time.
As a professional, my experience of GeneLab is spent off so much. I never have worked with space biology before, never. The data I'm having access now and the kind of ways I have to think now It's so multifactorial and so related to physics and metallurgy and biology and statistics and how to put everything together in a way that makes sense. It's making me a better scientist. So when I look into the data, I look into the data without any expectation and without any prior knowledge of what could be causing that. So when you put people from all around the world with all different backgrounds, they're looked at the same data as you, and you don't know what they're going to see because they have different backgrounds. So the potential of GeneLab project or making this kind of research to go faster and broader are gigantic. And this by itself is wonder.
I love that stuff. You know, that big hand, broad hand wavy stuff. So that's my mate William. He's over in Europe. He's based in Ireland, but he's from Brazil. Truly international collaboration. He does a lot of good work with mice. Yuck. So we've got 238 studies currently available with data for you guys to access. And this app is designed to help you find a study that you think is interesting. And there's so much metadata, but I've managed to simplify down to these three things: factor type, assay type, species. And then below it, we can display the data as like bar plots or as like a pie chart. You can kind of play with those to find a way that you think is easy to interpret the data. And then on the side here, we've got this little thing that can pop open. So this is the name of the GeneLab code for the study, the name of the study, the project title, which is kind of the same thing, but NASA likes to be redundant, and links to some extra metadata associated with this study. Okay, so obviously I'm gonna choose my favorite model organism as Arabidopsis, because to do anything else would be just a terrible idea. And then I'm gonna go, okay, my favorite technologies. Okay, what do we got here? We've got RNA-seq, mass spectrometry, DNA microarrays. Well, let's go for the most common ones. Okay, I'm just gonna filter down to DNA microarrays and RNA sequencing technology. Okay, factors. There's a bunch of factors. Oh, they're all so different. Oh no. You know what? This is the worst part about the GeneLab metadata, and this is part of my campaign to make the factors be a bit more like logical. But it's okay. So I'm gonna go across. Assays and protocols. Okay, so transcriptional profiling. Things I selected were transcriptional profiling. I'm just gonna click off that. So I've removed those filters that I tried previously. Okay, cool. Trying to put the Arabidopsis one back on. So I've changed my mind again. Arabidopsis, sure. Go back. So I'm just playing with the data here, trying to make a decision as to what study we're going to look at next. I'm going to stick on to transcriptional profiling as my assay type, because I don't really care if it's RNA-seq, I don't really care if it's a microarray, either will do. And there's multiple different types of microarrays. You've got Affymetrix brand, Alliant, a few others. Okay, cool. So let's move along. I'm going to go to the next dashboard over. What's this one? Protocols. Ooh, material type. That's a good idea. In fact, this is part of my campaign because it says undefined. So that means in the NASA GeneLab database, they have a bunch of studies where they haven't defined what material they harvested. Isn't that terrible? Look, here it are. These are cell cultures. So I'm now I have contacted the GeneLab team, and I'm in the process of helping them update their metadata, because this app really finds the gaps really easily. Anyway, I digress. So material type. So which part of the plant are we going to harvest? We can go for seedlings, whole organs, roots, shoots. I see multiple roots. There's a root. So there's more than one root. Okay, there should actually be more than that, but again, there's these gaps that they need to fix. You see, look. But this is their integration. I've just created an API to pull their data straight off their website. I then So when I make my own ones, I have to like fix these gaps. So let's go to this thing here. So roots. Okay, we've got 2 root studies. So I could compare the data from these 2 assays. Interesting. Okay, so 121. If I had pen and paper, I'd write down 121, but it doesn't matter. So this is me just kind of exploring the metadata as I'm trying to find a study that has conserved features with other studies that I'd want to explore. Okay, so maybe I've selected based on Arabidopsis. Okay, if I now wanted to download this, I could just click on the green button here. Oops, click on that green button there. Where's it gone? Well, usually when you click on that green, there it is. It's just appeared there. You can then click on that, and I can just download this table. So, and then I could have my metadata. Usually I do that, and then I'll go into Excel. So this is the microbial version. I've just pulled down the data and put it into the spreadsheet, and now I'm trying to point out the mistakes that NASA have made. And so I'm sending this documentation back to the GeneLab headquarters, and we'll be getting them to fill the gaps and fix the inconsistencies. Doing the same thing with the plant study just here. So anyway, so but the moral of the story is this app here that I'm just going to close down now. These apps here and here are interactive and designed to help you go through and filter through the massive GeneLab repository. They've got 350 studies. There's a lot to choose from, and a lot of the data is buried in below, and so that's why I made this filtering system here to try and help you find interesting studies. I went through that really quick, but the moral of the story is you should play with that. And when you find patterns, you can download those, you can take screenshots of the pictures, you can copy them and download them, and you can save them into a slideshow. And that helps you to create a story as to why that study exists and why that study is interesting. And you remember, as I was going through that, I said 120, didn't I? GODS 120. So as I was going through, I found a study, now I'm interested in it for a reason, and I can kind of go in and have a look for that. Okay, cool. So that's like the metadata app, and that's to look across the whole of the GeneLab astrobiology data sets. There's a lot of data there, and if you want to get to use the app, if you want to get used to it, you could probably just look at the plant stuff in isolation to begin with, because it helps us remain a bit more focused. So again, I've created this metadata app to kind of look through the plant data to help you find data sets. This is a bit more advanced now. To help you find data sets which are links between multiple data sets based on the metadata. So another game to play with, GODS 7, kind of fun. Anyway, again, so now we're going to go, so once we've used these metadata apps to find a study or two that are interesting, we can go in, the tutorial page, I forgot about that. Is this any use? And then we can actually go into the actual TOAST app itself. This was the first version of the metadata app that I made, and that's related to the paper and is discussed in the paper that you guys read. So you read some of the results that came out of that. You can go in here and poke around again, find links to the metadata. But now we're going to go into the actual genetics app itself. So TOAST was made for GeneLab, and so we're thinking of going to Mars and how space biology will change the way we look at life on Earth. So I've developed some codes to suck in the data from GeneLab. This particular app has about 45 of the studies from GeneLab's 250 odd. So it's quite large, but it's a bit more focused because it's just looking at the plant stuff. And so you basically have a menu of transcriptomes and proteomes to look at. And then on the end here, some of those studies are terrestrial studies that kind of might be linked. So terrestrial studies related to hypoxia, like CO2 in the environment, and the plant's response to reactive oxygen species. Because previous studies have shown that some of these terrestrial stressors might have some kind of similarity to some of these space-related studies. There might be some sort of overlap. And so these terrestrial stresses might tell us something about the stresses of spaceflight biology. Okay, cool. So we're going to go in, we're going to start to play with dashboards. Dashboards will basically function the same, but I like to have KPIs, key performance indicators, in a corner. So this thing here is counting how many genes are currently being selected by the database at that moment. And then those genes that are selected are shown on these graphs. And then the function of those genes is then displayed on the side here. And then like the statistics can be pulled in. To understand all the genetics, I recommend reading the research papers, but as I mentioned, 250 is a lot to get through. So that's why the metadata tools are designed to filter into 1 or 2 studies that are interesting. These dashboards makes that genetics available, but to make this genetics data make sense, you got to research how the experiment was conducted. What was the factor that was being investigated? How was the experiment designed? And then you can pull down the final list of genes, and that gives you your genetics. You can then start to explore what those genes do, give those genes character, treat them like individuals, and then try and understand what they're doing, because what they're doing is going to tell you about the environment. And we want to ensure that our environment is healthy for the astronauts to be in. And so understanding how the plant's responding to the environment will tell us how we might need to adapt the environment to make sure it's healthier. Volcano plots are great ways to display genetic data. They show both the fold change and the p-value. Genes going up in expression in spaceflight, or genes going down in expression in response to spaceflight. The higher up it is the more significant it is. Normally your p-values are small. The smaller the p-value, the more significant something is. So we do a negative log 10 transformation of the p-value to make really significant genes have larger numbers and kind of jump out of the pack. I like to have significance filters, so I can just press yes, no, and just remove the genes that aren't significant from the dashboard. Sometimes you want to search through the database based on the gene function, and that's what these buttons up here for. RNA-seq studies, you have absolute measurements of gene level expression on Earth and in space, and so that's displayed down here. But you'll see on some of the microarray studies that these data are not available. Once you have a cluster of genes selected, you want to know what it does. And so we do what we call genome ontology analysis. So that allows us to do a significance test to see if any particular groups of genes are enriched. We tend to do that significance test in string, but to make the exploration of the database faster, we remove the statistical test and just do gene counts in this system. So we're counting how many genes are associated with a particular genome ontology term. So genome ontology is a function of a gene. So genes are known to be function in certain places in the cell, like the cellular site for ontology. Some genes are grouped on a gene ontology associated with their molecular function, and some based on the biochemical processes they're involved in. That's the KEGG pathway. And so if we can find small groups of genes that are doing interesting things in orbit, we can then start to assign function to them and give them character as a group before giving them characters as individuals. All the figures are fully interactive, and so you can explore it quickly. Okay, cool. Let's play with the app. That's the description of it. Okay, so I'm just going to go and click on this button here. Okay, cool. Ooh, I made this video. I'm going to turn off the sound. I'm going to go big. Okay, so this is a video that I made of me playing with TOAST app when I was in the airport waiting to fly to a conference. So I made this cool video. It shows you that this is part of the journey to Mars, the most exciting journey that humanity will ever go on. At the moment, we're using the International Space Station and low Earth orbit to understand how life is going to respond to microgravity and stresses associated with spaceflight. Our goal is to improve veggie production in orbit. So if you can select an experiment in the TOAST Explorer, you will be able to explore these data that will help us go on this journey. We're going to select a study maybe based on reactive oxygen species, because ROS is known to be associated with spaceflight. And here we're going to do, we can see the number of genes that are currently selected in the database, and we're going to have to select a subset of genes. I'm just going to use the lasso tool, and now it's activating a bunch of genes that are activated by the addition of hydrogen peroxide. So now we just have a small fraction of the overall genome selected, and we can start to assess their molecular function or biological function or their subcellular site of function. There's many different places in cells where proteins are known to function, and we're just going to select the mitochondria because there's been some papers that recently discussed the mitochondrial response to spaceflight. We can now go look at another study. So I'm going to look at GeneLab's dataset number 37. This study is an experiment known as BRIC19. So now we're looking at the genes that were selected in the reactive oxygen species study and seeing how they respond in spaceflight. So now I'm going to select the significant genes, so the ones that responded significantly in that study, and we can see the overlap. The goal is to grow healthier plants in orbit so that we can ensure that we can colonize outer space and live and prosper. Cool. And that whole time, if I wasn't talking over the top, you would have heard my terrible music. Okay. So that's just a 2-minute thing. You can just go in and play with it. So again, I'm just in the normal viewer here. So this is the menu that's on the website. So the goal is to go in here, select some genes, pull them down, and then put them into these tools here. Because I want to do an exploration of the genetic network. So these are a bunch of genes that responded to spaceflight. And by mining the databases, I found links based on the protein structure and the papers that they were cited in that looked a bit like that. And so I want to help you guys explore this knowledge network to find patterns of interest. These tools will allow you to go to the next level. So I'm going to press Command and click on String. So your goal is to find lists of genes and to be able to put those lists of genes in here. And because this will help you do statistics to describe what those lists of genes are doing. Shiny Go, I'm going to click on that, is the same deal. If it loads up, which it didn't last yesterday, so I might have to try and check this link. Oh, no, there it is. Again, you can paste a list of genes in here, and it's going to do a statistical analysis of the list of what those genes do. Netminer, similar deal. This one here, you create a list of genes from Toast, and then you come to Netminer, you go to Arabidopsis. It's going to load up in a second. And then you can paste your list of genes in here, and then keywords in here. Mine's already suggested a bunch of words that are of interest to me, but you will have to come up with your own words of interest. And this app will then mine the literature to create a network that's a bit like that. But you will be able to go into that network and explore it and click on the different links and find different things. Finally, I want to bring your attention to this tool. This is a Venn diagram list tool. So maybe you'll go in there and you'll find a set of genes of interest. But then maybe you'll find another set of genes. Maybe we're going again and again, and you have so much fun that you're going to want to compare the different lists of genes. You just paste them in here, and this shiny app will make you the Venn diagram comparison. Load example. And then you'll be able to download those figures and those lists. And then you can do the research yourself. You can start to practice building slideshows that are designed to show people how life responds to outer space, a bit like this. Okay, cool. So, but it all starts with small steps. Okay, so Venn diagrams, etc., etc. So, okay, I'm just gonna pop this out into a new tab because I don't like these empty bits on the side. The user experience is very important. Okay, cool. I've already shown you the introductory video 1. Introductory video 2 we'll look at a bit later because this one shows you how to design an experiment to send it to space to test the hypothesis. But now we need to build a hypothesis based on previous spaceflight biology. This is one study from that menu. You can see the structure of the dashboard is similar to all the other videos and things I've been showing you. And so I've tried to maintain that structure throughout. Oh, I remember something. We played with the metadata earlier, didn't we? And we discovered there was a study that was of interest. Remember? So now you see why we play with the metadata. It's because sometimes there's so many different studies to choose from. You need a reason to be interested in one more than the other. And so we've already learned something about this study. And can you remember what that was? In the metadata, said it was the root. So we know there are other studies where they look at the root. So by looking at this study, we know that in the future, we'll be able to, whatever we learn about this study, we can compare and look at those other ones, because there's that metadata feature that's conserved between them. Okay, cool. So we're really going to want to understand this study more. So I'm going to go straight to the paper. This was a study that was done by Rob and Annalisa Poole from Florida, when they look at the plant response to spaceflight. And they're actually going to be looking at the roots of the plants. I'm just going to scroll down, skip the literature. So here's the pictures. I'm very much a pictorial person. You can see what they do is they sellotape, or they not sellotape, they use Velcro to attach the Petri dishes to the wall of the space station. Isn't that awesome? So it's very simple, but I like it because the Petri dish, square Petri dish format, is commonly used as a growth environment for Arabidopsis on many studies on Earth, in fact, nearly all the studies on Earth, and also a few of the studies in spaceflight. So this is one of the reasons I like to select this study, because one of the metadata features is conserved and is throughout many studies. We have photos of the plants, and I've actually accessed the raw versions of these photos. They're stored online, and I've reanalyzed them myself to get a few extra features out of them. So I've got a slideshow of that hidden away somewhere. In this study, they just measure one feature, the root length. I've measured the root width, the waviness, curvature, a bunch of other stuff, because I'm such a nerd. And they look in and they find some genes respond. So there's an extra metadata feature here. We have light and we have dark. Extra metadata feature, they have Columbia, WS, and Phi-D. So Columbia is the wild type of Arabidopsis from Columbia, Missouri. WS is another wild type of Arabidopsis from Warsaw, something I can't pronounce, in Poland. So this is your Polish variety. It's similar but different to Columbia. And so we can use natural variation to explore what's happening in many environments, and in this case, in spaceflight. I'm a geneticist, so I like to do genetics. Phi-D is a mutant plant, and I think it's in the Columbia background. So it's a mutant plant that has an altered capacity to perceive red light. Phi-D, so it's phytochrome D. Phytochrome is the red light receptor. I think. It's been a while. So anyway, so here they've got on these nice heat maps kind of summarizing the genes they discovered as being up and down regulated. Great. Some of this stuff involved in cell walls. Okay, cool. So I'm starting to understand their data, but I like to fish, and I'd like to compare this to other studies. But as you can see in their paper here, it's not really interactive. I could download the tables and fish through it all, but that'll take a lot of time. So I've done that for you. And then put it here. Okay, cool. Dark grown, light grown. Ooh, what's it gonna be? Dark versus light. Dark versus light. I'm gonna go dark. Yes, I am eccentric. Okay, cool. So why did I choose dark? I flipped a coin. Or I could pretend that there's other studies where they grow plants in darkness. And so this would give me a metadata feature to compare it with another study later. There you go. That's a better reason, isn't it? Using logic instead of just flipping coins. Okay. So genes significantly changed in darkness in Columbia. If I go over to the next one, this was that Phi-D mutant. And one of them, I think the WS ecotype. There it is, WS. So can you remember in the paper, there are the three different varieties that were compared with the Venn diagram? Well, I've separated them out because you can have too much information on one dashboard. Okay, cool. So I'm gonna stick to coal. Columbia is my favorite wild type plant. It's the one I do the work in my lab on. So I'm gonna focus on that one. So I'm just gonna select the genes that went up. I want to know the genes that were activated. If this was the case, I truly did just flip a coin. You could also talk about the genes that are suppressed, but I wanted to go up. Okay, so I've got my drop down menu here. Oh, I could go and look at another study. Actually, no, I'm gonna come back here. I could, no, there's no microRNAs in the studies. That's the thing. None of these genes involved in the methylation study that links to it. So genes that were methylated were found to be methylated in this particular study by Xu et al. I could start to search for specific gene families. let's not do that for now. I could look for the transcription factor. Ooh, RAV. But RAVs and Workies are my new favorite transcription factor families. Let's use that today. Oh yes, that's fitting. Follow the whim of the day. Oh no, I can't do RAV and Worki at the same time. Yes, I can. Ha! This is awesome. Okay. FYI, Worki for 22 is my new favorite transcription factor. Because my friend Arco is doing some lovely work on it. So these are the genes, the significance. I'm looking for significance. Oh, look, it responded significantly. Isn't that nice? I knew it would. Okay. So over here we've got, so we're using sorry, I digress. You have to enjoy yourself, right? Actually, so this is research happening live. So we've selected these transcription factors that we know are involved in stress. The RAV family are known to be involved in flooding as well, like low oxygen stress, which is a feature that has previously been linked with the spaceflight environment. These are the biological processes that are known to be associated with these genes. Some of them like response to bacteria, but other ones down below here. So basically, I'm now going to try and find out the function of some of these genes. Okay, go over to molecular function, have a look at that. Ooh, okay, this makes sense. Okay, so I remember I put the transcription factor filter on. So it makes sense that the biological function of some of these genes would be transcription factor activity, right? Or like DNA binding, because transcription factors stick to DNA. So this is an interactive relational database. So as we move through it, the filters that we put on have a consequence, right? So we'll close that down. Subcellular. So again, I switched off the transcription factor filter. I went back to the broader sets of genes. Actually, I'm going to go back to my favorite two transcription factor families. I'm going to go to subcellular localization. Nucleus comes up. That makes sense, doesn't it? Like transcription factors function on DNA. DNA is in the nucleus. If we stick on transcription factor filter, we're going to select for the nucleus, right? And vice versa. Ooh, ZAT12. That's a reactive oxygen species responsive transcription factor. Interesting. Which is a feature associated with spaceflight. Okie dokie. So all this time, we've just been looking at this one study. So instead of using the function of the genes as a filter to look at subsets of the data, instead, what I'm going to do, I'm going to go look at a different study. So this was wild-type Columbia grown in darkness, looking at the roots. Open up. These are all those studies. I could use the pictures to select for the a study which is similar, because I drew these pictures. Well, I made my friend Kai drew these pictures to represent the metadata. So astrobotany.com, go look at the good educational materials there. Also got some awesome cool t-shirts that he likes to sell. But you can also change the way the menu is presented. So if you don't like the pictorial form, which is great for me, who is someone who is one second. Hi Nitin. I'm gonna have to call you back in about an hour. I'm doing a teleconference with Carthage at the moment. Is that okay? I've got terribly sorry, mate. I'll speak to you soon. Sorry. Thanks for calling. Okay, so that's Project Mango, next stage. Okay. So, oh yeah, so I'm going to change the menu here to be this long one down here, going down. So now we're scrolling down. We wanted to look at something in darkness and roots. Oh, my favorite study. Okay, so GLDS 37. This is this first study I helped Professor Gilroy do on the International Space Station. And we grew Arabidopsis Columbia variety, so it was the same variety from the previous study, in orbit, also done in darkness. So a second filter has been applied. And last time we selected for the genes that went up, didn't we? So let's do that again. Okay, cool. So now we've selected a set of genes that went up in 2 studies, both when the plants were grown in darkness. And we've just used the statistics and the fact they were upregulated. And we got down to a list of 18 genes. And we can look through here and see what they might be involved in, like jasmonic acid and ethylene related like signaling. That's actually really interesting. I like those areas. And we can actually make this go large. and we can save these images. You can embed them onto websites with this code, so you can make a what's called a web document. So this is an iframe. So you can use iframes to if you make a website like using Google what's it called? Google website software page. You can just embed these into your own website. Anyway, and you can also take screenshots of them.
Quick question. So from this bar graph here, could you select like cellular process as a filter and see what is that 3 genes that are involved with cellular process?
Oh yeah, totally. So you can come in here and you can click on any of these things here and select subsets. To go further, I'm going to press yes. That's a great question. And now that's been applied as another filter. So it depends how small you want to go here. Or maybe you should use statistics to select what you do here. So what I'm going to do, I'm going to click on this green button. Click on it again. There it is. And I'm going to click on this thing here. And I'm going to click on this button here. And I'm going to save that. Ooh, and then I'm going to open it. Yes, I'm going to trust it, even though it's telling me it's unsafe. And now I have that list of genes, and I have the expression level of that gene on Earth. I have that expression level of that gene in space. And I have its, what else do we have? Its p-value, its q-value, which is its adjusted p-value. On the bottom down here, we have the filters that we applied. This transcript ID, this selection thing here, we have to remember, again, I did do that manual selection, so it doesn't have a name for that thing. So you'd have to type in upregulated. But this is the scientific process. You want to record why you got interested in these genes. Okay, cool. So I'm going to select the locus ID from that. Copy that, minimize that. And I showed you some of those tools earlier, right? Let's go for this one. I'm going to write these key space flight terms. I'm just going to click on the search button. So this is now searching through the genetic databases. I'm going to select the genes or links and perhaps some that are attached to them, and I'm going to view that network. So I want to have a look at all the literature associated with this list of genes. And so it's just going to load up that network there now. I could put it in here, paste it in here. So eventually you'd start comparing this list of genes to other lists of genes. So you could put it into this tool as well. Right, so this is, we'll just leave that there for now. This one is still loading. It's being a bit slow, but that's okay. What was the other tabs?
Can you show really quick again? Sorry, how did you get up the Excel spreadsheet? I didn't see the buttons you were clicking for that.
Oh, yeah. Okay, so there's this button here, the green one. So I clicked that, and then I clicked on it again, because the first time it didn't do anything. Second time it did. And then it produces this green button just here. And then you can click on that one, and that's your download. And then you just click on XLS, and it just pulls down. You can put it in a Word form. That's weird. And then, but the only reason I did that really is, oops, that's the wrong file. Where did it go? The only reason I did that was to pull it. It doesn't even matter. Was to get that list of ATGs, that list of gene names, gene identifiers. Whoa. Okay. Which I then just pasted straight into here. But that Excel file you would hopefully develop, or maybe you would move it into a Google Cloud form to make it easier to share. That's what I tend to do. But then I just very quickly got this list of gene identifiers off it to paste it into here, because this is going to tell me a little bit about how those genes they interact with each other. Whoa. Okay. So on the bottom here, we have loads of genes that are linked to them. Whoa. So one of these genes is really freaking sticky. This guy here. Whoa. Okay. So why is this guy show info? I just right-clicked on it to get that to pop up. So why is this gene so sticky? It's going to pop open to a new tab. So I'm literally, this is how I do my research. Encodes a protein who's similar to tobacco hairpin-induced gene, non-race but disease resistance gene, NDR1, expressions gene and mosaic virus. So this looks like it's a pathogen perception gene. So it's sticky as hell, okay? Sorry, I got them. But this is the nature of research. It's about finding a gene that's worth mutating to investigate. And that is a gene that's worth mutating to worth investigating, because if it sticks to lots of stuff, it might be important. Jazz1, that's cool. This is, again, it's a highly interactive node, links to lots of stuff. It's involved in plant response to pathogens. Because pathogens would be interesting because the spaceflight environment, there's many papers that have shown that your immune system is altered and the plant's immune system is altered. We've seen plants succumb to pathogen infections in spaceflight. So understanding the genes that are responding in spaceflight, particularly those associated with pathogen response, is like interesting. What's this one down here?
So just a quick question. When you say the sticky genes, are these specific genes that are related to the list you added to this
software?
Yeah.
Exactly that, yeah. So so this list of software in here, this list of genes here, is these yellow nodes, these ones that are highlighted in yellow. The ones that haven't been highlighted in yellow are the ones that are known to interact with those yellow nodes and also interact with any of these keywords.
Okay, that's what the word search is for. All right, thank you.
That's cool. And it's, yeah, again, feel free to like fire those questions anytime, mate. Literally, I'm just exploring the data, just kind of showing you how I use this ecosystem of tools to maximize the impact of this spaceflight data. Because we did like 2 little comparisons like this, and that's what the TOAST paper is. You see in the TOAST paper, we go into the mitochondria, we use that as a filter, and we find some and we look at the study associated with the Shenzhou spaceflight. Shenzhou, is it down here somewhere? From the Chinese Space Agency here, these studies here and here. So we compared this study to this study and applied the mitochondrial subcellular localization filter, and that was the research paper there. But as you can see in this software, we have got a lot more data sets. There are so many patterns in here and so many stories to find. And this is literally how you do it. It's really not that difficult. Okay, so
Richard,
here's the next one. I'll go for
one question. I think can you go back to NetMiner? Yeah, when you were selecting like a single gene in that map below, did it bring up, I thought I saw a picture of like expression of where in the plant that gene was. Like you had the leaves and the shoots and things somewhere. Can you show them that? Or was that a different, this one? So how did you bring that up from the NetMiner gene?
Good question. So I just right click there and then show information on the side here. So as you click on these different genes or different other nodes, they have information on the side, but sometimes it's not showing. But anyway, so I'm just going to click on this Worki. And it's got the descriptions here, they're based different names. It's been researched by so many different people. It's got 6 different names. Great, that happens a lot. But there's a unique code, which is an identifier. This is the TARE identifier. Arabidopsis, Thaliana, 2nd chromosome, gene location is this coordinate here. That just popped open into this new tab viewer here. And then, oops, daisy, it loads up all that genetic information about it. So this was a different one to the previous one. This is Worki 33. It gives you a quick description of it. It shows you its gene structure. So we have exons encode amino acids, got introns that get spliced out. You can download that sequence if you want to start doing genetic engineering with it. That's where you start designing your primers. And then here we have this data source. So these are a pictorial way of observing where this gene is expressed. But you know what? There's a better way to do this. There's a much more beautiful way to see these exact same data. And that is called ePlant. I used to have ePlant hidden inside TOAST, but I had to remove it because they changed their security processing and they don't like being inside TOAST. Damn it. So I'm going to type in So, okay, it popped out of here. You saw that actually, but now I'm just going to paste it into my favorite tool, ePlant. So ePlant allows you to take a single gene and give it character. You can start to get to know them one-on-one. I was going to do the group statistics before delving into this, but you can see here it very quickly starts to give it character. So Worki is a transcription factor. It's obviously going to be in the nucleus. It's conserved across all different like species. There's natural variation in expression. You have different varieties from Arabidopsis from different places around the world, and some of those varieties express them more highly than others. The gene is probably expressed in most tissues, yep, but it's also more highly expressed in roots. I like root expressed genes. I spent a lot of time working on roots. That's boring. But it's not just the location in the plant's aerial tissues. You have to think of the embryos developing. You can look at the shoot apical merostems and start to see about where your leaves are starting to develop. This is where it's in later life. But I'm more interested in roots. Roots sense the environment, sense gravity. This gene is not in the gravity sensing area. Okay, that's cool. But it is in the stress responsive area. These cells just here. And so does it respond to stress? It looks like it responds to UVB. Oh, wow, look at this. It goes up massively in response to solar. Interesting. Over a timeline experiment. So we're giving it character. So now what I'm thinking is this gene is a stress response gene. It's in the nucleus. It's a transcription factor, regulates the response of thousands of genes. If you add salt, eventually this gene comes on, so it's activating the long-term stress response to salt, which is increased levels of background ROS, which is interesting because that's what is the short-term response to UV. If you give a plant UVB stimulus, you get a short-term ROS production, and this gene comes up. So maybe this gene is a ROS response gene. Maybe. Anyway, so this network. So yeah, so you can give individual genes character. Once you find a node which is of interesting, it launches straight out from the side here into TARE. TARE is the Arabidopsis information resource, but ePlant has been made by the University of Toronto to explore the TARE data in a more user-friendly, more aesthetically pleasing way. One last thing about ePlant. Where did it go? There it is. With ePlant, if you really get interested in a gene, you can do some art with it. This is artistic. You can look at its protein structure, and you can look at its different domains. This is the bit that makes it a Worki. You can see if it interacts with other proteins as part of an interaction network, and then you have to start working out what all those other proteins do and describe the function of this network. And then if you think that gene is worth exploring, you can come to the genome viewer. You can go straight down to the location on the genome. This is those exons and introns that we saw earlier. But if you come to the genome viewer, you can also see where there's mutations in that gene. This is Sulk-seq. Sulk is a center where they mutate Arabidopsis, and they have got mutations in this location and this location. And so that mutation might prevent the function of that gene. So they can be ordered, those seeds can be ordered from the internet. There is a link out by right-clicking on that. TARE DNA order from ABRC. So this stock name here, this germplasm is your seed stock name. You can get that ordered, and then you can genotype it. So you can try and check if that tDNA insertion is there. So these mutations were created by Agrobacterium. Agrobacterium inserted tDNA, transfer DNA. Agrobacterium transferred tDNA from it to the Arabidopsis randomly and made some mutations. And hopefully that mutation has prevented this coding region being expressed correctly, creating a new allele, which might have a phenotype. This Worki gene has been shown to be involved in salt stress and pathogen response. Wouldn't it be interesting if this gene is also involved in plant response to spaceflight? And that would be important for NASA to know, because pathogens might be present in spaceflight. Well, they are present in spaceflight, and that's in the MANGO database. You can find out which fungal pathogens are present by looking in MANGO. So understanding the function of this gene might be really important to the future of plant growth in the spaceflight environment, if we want to do so sustainably.
So I have a quick question. Sorry to interrupt again.
No, anytime, mate. Go for it.
So this ePlant program, the level of data they have and like specific, at least for this gene, is crazy because it looks like they even have like the structural function of it. But my guess is that this TARE database has so many Arabidopsis genes, and we don't know the structure of all of them. So if you go from each different gene, are some of them going to have like more data than others? Because like, for example, yeah, like the test where it had all the over time for a bunch of different response factors. But does every gene have that, or is it kind of hit or miss?
That is a fantastic question. So in this database, most of the genes that are in ePlant have the graphical representation in these heat map viewers and the various other ones. But you are right, there is a gap in the system, and that is best illustrated through the difference between microarrays and RNA-seq. So not all of the genes are in there. You are totally correct. Microarrays only assess a proportion of the genome. So when we put these plants in spaceflight, we use RNA-seq. So we have the genome had been sequenced, so we were able to look through and see every gene all at once. So if we select this, we can select all the genes all at once, and this gives us a rough count of the number of loci that are on the Arabidopsis genome. There's multiple transcripts, so more than one gene. So one gene can encode more than one transcript, and that's why we have all these extra like gene IDs, as we're calling them, but lots of these are transcript variants. What I'm going to go to now is a microarray study. So this is a microarray study. Now I'm going to select all of the genes in this microarray, 18,000. So you see there's less loci that are being monitored in this study. That's just the nature of the technology. So when we look at ePlant, actually I should do one last thing. So selecting the microarray. So we're looking at, and the microarrays were selected carefully, so most of the important genes are on the microarray. But when we look at ePlant, this is microarray technology. So now you see the initial biggest gap that's there, right? So then when you go into TARE, so TARE is going to have gaps here because TARE has every gene, but not every gene has an ePlant data set for it.
Yeah.
So yes, you're totally right, and good point. Well played. So yeah, okay, cool. So net minor, so we had that list of genes, and I'll just place them in here. So I want some statistics. Well, I don't, but my supervisors always do. And you should too, because if you, cancel that. What have I done? If you're going to focus in on a subset, you want to use some stats to do it, because there's a lot of options. So sometimes it's good to use stats. And I'm searching for Arabidopsis thaliana. It's got to be in there somewhere. There it is. Select that. Search. So this list of genes again was the ones that were selected based on 2 spaceflight studies. You always have to remind yourself because you do this. 2 spaceflight studies, both done in darkness, both done in Columbia. Wow, this is a really nice network. Actually, I'm not joking. This is both aesthetically pleasing and insightful. Okay, if I stick that out there, I can put that in there. Okay. Okay, it's going to be the top corner there. It's going to be there. It's going to be there. Okay, I like that better. Okay, cool. Jolly good. So, in fact, here you can see, this is a great example of the same principle. Can you see these pictures in the middle here? That's showing actually their kind of the picture of their like molecular like structure. And so these ones here don't have that. So yeah, that's another great illustrator of that principle that you were saying earlier about the inherent gaps in the system. These lines here are connecting these genes. Just have a quick look down here. We're doing some statistics below. So first things first, settings. There's 2 little options you can change. Static. Yeah, so these are being linked based on the evidence, or it can be the molecular action. So I'm going to click on the interaction there, and it's going to tell me a bit about the connection between those two genes in the network. F-box, proteins, interaction, co-expression, okay, co-authorship. So I'm not really too sure. I don't think these are the best connections, co-expression and co-authorship. There's not a lot of great discoveries been made based on co-expression. In co-authorship, there is prejudice in humans, but molecular interaction, we'll switch over to that and update. I like that better. Okay, so interaction. Hang on, where is that? Where's the...
So co-expression is just asking, are there studies where both of those genes were expressed together in the same plant tissue?
Yep. And some of them also respond to the same stress as well. So you do get the stress response in that one too. But this one here is better in my opinion. But again, it's all kind of personal preference really. So I do this so often. I get to play this game. Okay, cool. So still Worki. These ones here. So these are some sort of protein interaction. I forget which. Positive interactions. Oh, it doesn't describe why. I've never seen this before. Usually there's a colored line in one of these and it's describing what the interaction is. Maybe the only link is co-expression. Yeah, it's plausible. But anyhow, so, okay, Worki 37.3 and JAS1 are still interesting nodes, but statistics, this is why we came here, not just to play with networks. So this is the bit I enjoy. We're going to go to analysis. We're going to look at networks down here, okay? So now looking at the biological process, papers that have discussed them, biochemical pathways that are involved in, keywords, protein keywords, smart domains, PFAMs, so Workies, etc. Okay, cool. See, look, Worki has come up as being significantly enriched. On the side here, we have the false discovery rate. So these are our p-values, our significance tests. We can download all of these and we can stick them into our Excel file, our spreadsheet in the cloud, as we then try to describe the functions of these genes. These are important for your paper. Statistics has never been so easy, mate. Make the most of it. Okay, so biological processes. I really want to understand them. Holy golly, I didn't expect that to be so big. Okay, cool. Gene expression. Okay, cool. Blah, blah, blah. Water deprivation. ABA, stress response hormone. Jasminic acid, stress response hormone. What we got here? So this is the general stress response pathway linking to the general pathogen defense pathway. Huh, interesting. And you can select. Okay, so now I'm going to look at the number of genes in the sets. I'm going to select one which has got more, or down here. Then these are the papers. Some about Workies. Oh god, there's a lot of them. Holy golly. Okay. Pathogens. Okay, I'm going to just click on the plant defense. Okay, because I've clicked on that there. When I go up, we've now fished out the plant defense pathway. And oh look, some of them, they're all connected. I wonder what that means. Probably nothing. I'm just a pessimist. But okay, so you see the game. That can now be downloaded. That can be exported. You can download as a PNG. It's kind of handleable, and it can be put into a slideshow. I should really do that because this is actually really nice. Okay, just going to download that. So which was this? So this was GLDS 37 up, and this was, oh no, it wasn't GLDS, there was a typo there. Then it was GLDS, what was it, 120 up. So this is coal, dark, light. Best place in the world to save everything ever, the desktop. Yeah, right. In your folder in the cloud, you'd move that to, right? So that's how you start to fish through. So now, because of this network, why did we do this? Was it just to make a pretty picture? Yes. No, it wasn't. Was it to do some statistics to keep the bosses happy? Yes. No, it wasn't. It's because we have to focus. Sorry, like you can see my internal ADHD boy trying to not focus throughout that whole process. And now we have 4 loci to focus in on, right? So this is the fun part. E-plant. Okay, so we got one of them. Crap, what was the next one on the list? Oh, I wanted to get. So we did Worki 33, Jazz 1. Okay. So this is the fun thing about being a scientist. You get to play detective. You really have to. You know, in this modern big data era, you get to play detective, and no murderers are on the get on the line. 33 and 22. Okay, so which one did we have previously? We put in one of those 2. I forget which one it was. Basically, you can load them up one by one here, and then you can do a cross comparison of them. It's just taking its time to load. Because I had to do that thing down there. Okay, Jazz 1. What's welcome screen? Gene viewer. This was done after Worki 33 is one earlier. So now I'm going to add Worki 22 because that was in the network. And that's going to load in. Okay. What's next?
Hey, Richard.
Yo, go for it.
Maybe this would be a good time to give them like a 10-minute break.
Yeah, do that. That's
all things are loading and can come back.
Yeah, totally. I'll just load up this next one.
You guys want to come back at 1:50? Okay, everybody can get a break quick.
Woohoo! Then after that, we can like answer some of the questions and have the discussion thing instead, because this is just like how to kind of use the ecosystem. But I think that's the gist of it, right? I think, yeah, on the whole.
Yeah, I think that'll be helpful, and maybe we can go through some questions they have if they want to review any of the tools.
I think so, yeah, because they're the main tools, and these other ones down the bottom here are the kind of the same kind of thing. So I think I demonstrated how the tools were used. I think that section is done, actually, yeah.
Okay.
That'll do.
All right, we'll come back. You can take a break too if you need one.
For me, sure. Which should probably just be more plant genetics. That's my kind of break.
If you don't mind me asking a question during the break.
Oh, go for it.
So you've mentioned earlier that kind of like Mango is a tweak, like a redesigned version of Toast, and then there's another one for the mice and such. So is this just kind of like tweaking the data so that it fits the new thing, or is it kind of totally, or tweaking the database, or is it totally like revamping it so that it fits microbes versus plants?
So the Mango change is more different. Hang on, so how to put it? I'm thinking. Okay, so all 3 of them are relational databases, but the underlying data structure is slightly different. But I use the same software to make the graphical user interface for each. So I think I was able to deliver a similar user experience for each, with the concept with the KPI to give you count, like a track of how much of the overall database you're selecting, and then the concept is it's an iterative filtering tool. So as you go through, you've got this overwhelmingly large amount of data, and you can see the big broad patterns, and then with a bit of detective work and cross comparisons within it, you can filter it in, and that's where the story is. And then once you get down to that smaller list, all of them you can expand back out into all of the knowledge of humanity. But you have to go in to find something really small and focused, then come out of it. So with the plants, we're going into the genetic networks, we're going down to a few genes, and then we're finding what that network does, and then giving them all character, and then we expand out again as we get all of like human knowledge and on those genes, which is a lot. With the microbial version, you're filtering down through the different phylogeny down to like the species level, and then eventually to like the size of the community level is your final like determining feature, instead of like significant enrichment of expression like it is of genes. And with the microbial, it's a selection system. It's you're selecting for the size of the population and the increased proportion of the largest proportions. And then with that, the iterative filtering comes in, which is what are human perspectives? And the human perspectives are like, what are human pathogens? What are plant pathogens? And using that, or like the location of the study as the filter to sub-select a story. And then once you've used those filters, you've then got a location like the dining room table, you've got human pathogens on top of it, so you've got human theories and stories, and then you have the most prevalent microbes in that location with those features. Then once you get that small number of microbes, you then have to come back and go into all of the human domain of knowledge and go, what's the story about those microbes? What can we learn from all the other studies done on those? So the way you use them conceptually is the same, but it is different data. So the user experience was made in the same software, so it looks kind of similar, but there is that slight difference. Does that make sense?
So then, yeah. So just to give you, I'm a computer science minor, and I've spent a lot of time working with databases. So I'm just kind of interested in a little background of how you're making this. So do you see like a human version of this up next?
Yeah, well, the human data is already integrated into the mouse version. So there's a version where we're taking the data straight in from GeneLab. They're pre-processed. I know we're up to like 80 studies so far on this. And we're just putting it in and linking it. And so then we've linked the mouse data to rat data to human data. And if you're interested in building like databases and like relational databases and such, we do have access codes. So you could come into the Gilroy Lab server, and you would be able to access these relational databases programmatically, in a way that you'd be able to make edits, changes, add and retract stuff. So if you're interested in that, again, shoot me an email. I can send you the access codes because I have, because it's the way to learn is to do. I use a software called Click. It's kind of based on what's it called? There's C++, there's some HTML. What's that? MySQL, SQL. Actually, that'd be a really important one because that's the area I'm really not that good at. So all the apps I've created have actually doing very minimal computing in like in the way I develop it. And with a little bit of assistance from people who know MySQL, there would be a lot more stuff that could be done with it. I have to build the filters in, so you go in and the user does all the filtering to find the stories. But I know you can use SQL to build some of the figures in a way that specifically shows those patterns pre-computed. But it's not my area.
Okay. I only know like very little SQL, so.
But that's how you learn these things. I knew none of this stuff. I'm a plant scientist. I did my PhD in plant science. Like, I had to come back to this whole computer stuff in later life, so to speak. And so one of the things I've learned through getting old is it's a kind of, you learn by doing, right? Like, you didn't wake up one morning and were a programmer. You didn't wake up one morning and were a biologist, It's, and I found like, I never, all the things I had to really learn, I learned by doing. And I feel like I don't really understand something unless I've done it. It's kind of, I'm a learn by doer. Some people can learn stuff from a book, lucky mothers. But so when as a kid, like I played computer games lots, but I never had to learn to program. Like Python wasn't even around. But then as I went through, you had to do something. There was a computer challenge in front of you, and so you just like you learn how to do that code to get through it, right? So that's just a hindsight thing. Yeah, does that make sense?
Yeah.
And that's why I love this concept of project-based learning. Like sometimes just the necessity of the project making you do something makes you learn something you never expected you were going to learn. And sometimes those things are more useful than the things that you learn for fun. Oh, hello. Anyway. Oh, so this is Baloo, my kitty cat. Well, one of them. There's another one somewhere. He's a good boy.
Baloo? Is that how you say it? What's his name after? Or
Jungle Book.
Oh yeah.
I've got a Disney theme. Got Simba over there somewhere.
No way, my cat's name is Nala.
Oh, so cute. Yeah, I think these cats are doing well out of this whole zombie virus thing, right? Are any of you guys in Michigan at the moment? No? Do you want to hear a scary story? So so they've tracked down patient what's called 0. So a friend of a friend, they were playing golf with this guy, and he's been recently contacted by the Center of Disease Control, and they were like, you went to a wedding in Wuhan in December, and he's like, yeah, I did. And they went, you came back with a cold, didn't you? It's like, yeah, it was a pretty bad one, and then my wife got it. And they went, and then you flew to California and New York, didn't you? He's like, yeah, I totally did. And this was all in December last year. So this virus has been in Michigan, California, and New York since December last year. So you know how like China spent all of January like shutting down to flatten the line, and how we haven't done any of that? Stay indoors. Anyway, so this is one of the other reasons I'm showing all these things. I'm trying to show you broad data science, you know, like these interpreting the data is really important in this day and age, like more than ever. And okay, we're using the space, I think, because it's kind of cool and a bit frivolous. We're finding these networks and such, but essentially these same techniques, methodologies can be applied in other forms of science, and business and economics and all that stuff too, if you want to go that way. Yeah. So that was basically just how to use like TOAST, and that basic same principle of iterative searching and detective work on your end can then also be applied in the same way to the Cross-Kingdom Explorer and to Mango. And then, so the Cross-Kingdom Explorer, that's the one that links together the plants, i.e., so I take a Arabidopsis. Oh, actually, show you, I got some slides to this. You want to see the slides? Yeah, you don't want to see this little guy, do you? He is cute, though, isn't he? I'm going to go back to desktop 1, sure. And then you can do some of these questions as well, because questions and answers are fun too. But this is actually back to the first question, which is the mouse. So this was all about plants. Oh look, it's all loading up here. Once these things have all loaded up here, there is a way of doing an analysis of all of these that are linked. I forget what that is. It's one of these buttons up here. Anyway, I digress. I really need to close some tabs. So context, plants in space. What was it looking for? Yes, cross species. We've done plants in space. Because of the success of plants in space, we wanted to go cross kingdom. So can we look at the genetics of, say, a plant and learn something about how humans are going to respond? Why do I ask that question? Well, it's because NASA is so bloody human-centric. It's really annoying. So we have to play their game occasionally. Don't hate the player, hate the game. Oh, it's all loading up. Just give it a moment or two. It's fine. It's this thing below. This is now loading up the interactive tree of life. And the moral of this story, once this thing loads up, it's actually I might just change it to a pop-out thing because it, there it is. Cool. So that's the tree of life. Great. Okay. And so we can connect everything from like bacteria all the way up to humans with everything in between. If you do play with this, pop it out over here, and then you can go in here and you can play with the interactive data visualization and start to filter for something which is human legible. So this is one way to look at the tree of life, you know, and the reason it's there is because I do love the interactive ones. Here's a couple I ripped up from a paper. You can think of the tree of life as being like this kind of explosion, bacteria like here. So wow, lots of microbes. Or you can think of it like going back in time, like mammals, more advanced birds, reptiles, amphibians, fish, you know, going back to these conserved like ancestors back in the Cambrian explosion. But I like this one. Do you recognize it? Any takers?
It's Darwin's tree. He wrote in his notebook.
Good man, gold star, high five. Seriously, it's I like it. It's conceptually, it's to think like in order for us to build all of these things here, we needed huge amounts of data about genetics and size and shapes of things, and yet this dude just went on a few boat trips looking at stuff, and then somehow was able to envisage something which would be so profound. And the question is, can that type of thinking help us imagine what would happen in the future as, say, other planets undergo ecosystem synthesis when humans go there? Or perhaps converse, as humans cause ecosystem degradation, what will happen, what kind of patterns might evolve? So to try and get to the bottom of some of these questions, we imported a bunch of data from NASA GeneLab using an API system from not just plants, but this time microbes, Drosophila, flies, nematodes, which is microscopic worms, mice, humans, and rats, and stuff in between. And again, you're starting to see a similarity in the structure, hopefully, and how it's like a menu just waiting for people to explore it. The infrastructure is slightly different because there's just so much more. So I had to there's an overall summary slide, like section, which pops out into a custom annotation dashboard and all pops out into a custom specific dashboard for this study by itself. And that will make sense shortly. This is one of those custom specific studies for itself. So there's a huge amount of data that's ready to be fished through. And you can do that same thing where you can interact with it, do your iterative filtering to find a small subset of genes. Cool thing is, once you find a small subset of genes, we've got a very similar filtering mechanism to look at the function of those genes. I've done a slight summary. I compressed 3 graphs into one. In the TOAST database, we had the molecular function, the biological process, and the cellular components as separate things. Now I put them in here all as one just to make it quicker. And it's all linked. So here, so we've got a count of all the human genes have been selected, all the mouse genes, all the rats, the flies, the worms. So yeast are more primitives. They have less genes than these more advanced organisms. Similarly with plants, we've got like Arabidopsis and a bunch of other actual useful plants like tomatoes. And the reason they're selected like this is I actually connect their genes in the back end with what we call an orthology network. So as we go back through the evolutionary tree, some of those genes have a conserved ancestor and their sequences have been preserved to the modern ancestor. And so what we've gone and done is selected a human gene set, a human bed rest study, so selected all the genes that were significantly differently expressed in a human bed rest study, because lying in bed is a bit like not being in microgravity because your muscles start to waste because you don't use them as much. Get your exercise in while you're in isolation. And because we've selected these human genes, those human genes have conserved relative genes that have an orthology relationship in mice, rats, Drosophila, C. elegans, and yeast. And so, this network allows you to find the conserved patterns that transcends just like one organism. And we can show you how, because, well, in fact, you already kind of know how. I'm just gonna go back here. Little video showing the game in action that I made while flying to. I only make those videos when I'm sitting in the airport bored as hell, FYI. But this one, I didn't This one, I borrowed the music from SpaceX's favorite band. I didn't have my own electro music on that one. I guess that's just on Mango. Apps loading. These are great moments to twiddle your thumbs. Maybe do a bit of stretching. So I've got a new version of this app being made at the moment. This particular one, I don't recommend going on the mouse option because there's too much rodent data. So this thing here, when you click on it, there is like, I don't know, 30 studies. And each of those studies has got about, I don't know, like 30,000 data points, 20,000 data points. And it tries to load them all. So you end up sitting there for a while. So I don't recommend it. The next version of it, I'm splitting it in two. I'm separating the radiation studies from the spaceflight studies just to improve the user experience. Again, I've done the same thing where I've got a picture where I've tried to summarize the metadata. This one is a bit more minimalistic. One of the things we learned from the TOAST one was I was trying to have too much metadata on the picture and no one else knew what it meant because they didn't draw the picture, so they couldn't work out my art. Anyhow.
Hey Richard, is there any zebrafish data in there from the JAXA aquatic habitat?
Not in this one yet, no. Because I don't think it's been processed. Hang on a second. So the main determining feature is what's in this current version of the TOAST Cross Kingdom is which studies have been processed to a level where visualization is possible with statistics. So I've got a lot of extra bloody mice studies I have to add. Bloody mice. I don't think I've seen a fish here. I'm sorry to say. Oops. So I can blame GeneLab as the moral of the story. It's not me, it's them. But they're slowly working their way through all the studies. And so far you can see it's very mouse-centric and human-centric. Which, there's a fly. So you can see that's the problem with the system. In my opinion, because I'm not a mouse scientist. But yeah, they haven't processed the zebrafish yet. So that's why I haven't done it. That's that on there. So it's this app here. Yeah, I should probably close down the plant one because that game is done. Okay. So yeah. So the main selection criteria in this system here is that it's been processed by NASA GeneLab, so that I'm able to just pull in their data. So when processing high throughput sequencing data, there's an early stage of aligning the RNA fragments to the genome, which requires like massive amounts of sequencing power, massive amounts of computational power, and so that's really time consuming and difficult to code. So I'm basically waiting for GeneLab to do the processing and then just pulling it into here, and it's still a lot of work. Anyhow, so so I'm going to avoid the mice. Again, at the beginning, there's this metadata exploring app dashboard. I go through it periodically every now and then I go in and I pull in the NASA GeneLab data and I make a new way of visualizing it because I have to. But maybe I'm using this and I'm like, ooh, what to do. Okay. And then I'm trying to, so maybe I've picked a study based on logic, but I'm not going to use logic today. I'm just going to be random. But you should use the metadata to select a study based on a logical comparison that you want to do. But for today, I'm just going to go have a look at humans. Fuck it, why not? So we've got a lot of potential transcripts in humans. So, okay, 66,000. So it's a lot to take in. So I'm going to scroll down. Ensemble is a organization which has been describing the human, the genetic response of, Ensemble is an organization that's been collecting databases of genes and assigning to them functions. And so this whole thing is based on the Ensemble transcript ID. So that's the underlying genetic network that allows us to then link out to things like reactome. Reactome is a list of all the different like biochemical like functions and processes in plants. So this is the new edition. It wasn't in the previous toast. I pulled in reactome. Again, we've got the same kind of concept. You can use the cellular site as a filtering system or see where things are. And here we have our different descriptions, like the biological processes, molecular functions, etc. Now I can scroll down, and now we have all one by one next to each other. So just going to look at this first one. We can pull out the genes here later if we find some that we're interested in, but remember at the moment we've got a lot, so there's no point in putting this down. We've got the download option, it's there, you know. So now we're going to do a bit of this detective story work. So we've got hypergravity, human T cells, we've got lymphoblast cells, radiation, spaceflight, 3 or 4 radiation species. This one, the electromagnetic field, if you look within a study, you can find genes that respond, but if you go to it, they're not significant. It's go in, they're going to come out. Yeah, this study, just zoom into a few of them, selecting the most significant ones from within the study. You can see the coloring scheme here. I've set the coloring scheme. This is my subtle way of pointing out a study that's not significant. You can see how it's like this paley kind of gray area, and it's 1 to 1 here and here. So that's the adjusted p-value. So the moral of the story is the electromagnetic field of an organism is like a really mild thing, and so if you do a study, you can find genes that will respond to something as subtle as a magnetic field, but then you're basically just skimming the top few genes off the top, and it's pretty vague. So sometimes it's better to have a stimulus that is a bit more human GeneLab summary. Sometimes it's better to have something which is a bit more bold, shall we say, not just the background. So we're gonna ignore that one, FYI. Took me a while to work out what that grayness meant when I did that. Like, when you're going through all these studies, like, I don't do humans. Why am I having to build this? Okay, these cells mean something. What do you think they mean? Any takers?
They probably did it on a cell culture and not in like a live human specimen.
Spot on, nicely done. Yep. So you can pick it all up just off the picture, but if you couldn't, you could also click on it, pop out, and I'll take you to the GeneLab metadata, and you can go through that and work out all the cool stuff it does. A lot there, which again needs to be done to really understand the study. But low dose ionizing radiation, that sounds good. Because if you're in space flight, it's probably gonna be a low dose, right? Radiation, you wouldn't imagine a really big acute like source of radiation, would you? So go lab home, radiation. Quick look at radiation. So like, because when we're looking at this genetic data, so there was that low mild dose radiation, that's this one. Okay, but you can also imagine an acute, extreme high amount of radiation being added. So, whoa, Madison, if I zoom out from Madison. I didn't realize it was remembered Madison, that's weird. So, radiation in America, radiation in the world. Okay, so all around the world, there's always this like background, like pervasive radiation. In some places, there's more than others. So life has always had a bit of background radiation. When we're in our space, we're going up, we're really interested about intergalactic cosmic rays. There are other types of radiation in space. We have the sun ejecting like balls of plasma, which lead to like protons and flying through space. Whoops, daisy. And when they hit the Earth has this magnetosphere that protects us. And that creates what we call these Van Allen belts, as those protons get kind of bent around. Intergalactic cosmic rays just go flying straight through that like a massive, powerful cannonball going through everything. But the background cosmic solar wind from the ISS, from the sun, gets caught in these Van Allen belts, and it creates what we call the South Atlantic anomaly. this lovely video provided by the European Space Agency shows the lowest band of the Van Allen belts, and where it comes really close to the planet Earth. And this is in the near what's called near South America, near Brazil, and this is what we call the South Atlantic anomaly. And the space station flies through it. So there is a 40-hour, I think it is, periodic flight through this area of slightly higher amounts of background proton solar wind. Although intergalactic cosmic rays are the ones that astronauts talk about, the flashes of lights that they see frequency, as they go through their eyes. And we can generate similar things on Earth using like linear particle accelerators. And humans are starting to create new mechanisms to synthesize synthetic intergalactic cosmic rays, but that's still work in process.
Do we have any ways, sorry to interrupt, do we have any ways to protect astronauts from these intergalactic cosmic rays, especially once they leave the Van Allen belt, like the lunar missions or the Mars missions? Do we have any way to do it? None? Okay.
No. Some people would invoke praying, but I wouldn't. There is unpublished research from UW-Madison into the potential use of technologies developed to hold balls of plasma during fusion reactions, but turning it inside out. So basically creating the outside of a space station or a sphere within a space station, which has a special type of metal, which you can turn into a electromagnet, which is supercharged. And then essentially that might be able to deflect some of these galactic cosmic rays and just move them around it. You can't stop them. They're going to keep going, but you can like deflect them around the edge, possibly. Hopefully, we'll be able to test that technology in the near future. So you see how they're accelerating this with like a load of battery, a load of magnets, and you fire it along. What if you turn the outside of your space station into that magnet, that electromagnet? And then at the moment when that cosmic ray is coming, you push extra electricity into your inverted magnetic accelerator, you might be able to bounce it around you. So there's hope, but it's future technology. So, but in the meantime, the main type of radiation that is prevalent in low Earth orbit is going to be this oscillation of solar wind that's being captured by the Van Allen belts, and the moment when we get more of it going through the South Atlantic anomaly. And those reasons there are why I went for low dose ionizing radiation, because that's what I thought would describe that type of radiation well. So here, this figure here is showing us like the genes have the highest standard deviation along the bottom here. They're in that study hole. But in that study, there were 1, 2, 3, 4, 5, 6, 7, 8 different treatments of radiation that can be compared against each other. So what we saw on that first human radiation page is a gross oversimplification of a really complicated experiment. You see, they're using lots of different quantities of radiation. So I now go back. So now we go back to the Human GeneLab summary page. So I'm just gonna let that load, let that drop down. So these things here at the top, these are the summary pages. And on those summary pages, we have this one figure, which is a gross oversimplification of multiple different comparisons. Do you see my point? And so we can have this broad overlook here, but really you want to delve in deep and actually get to, into the belly of the beast. So it's the high dose radiation. Okay, so that's interesting. So maybe the high dose radiation would be a bit more like the intergalactic cosmic rays. Okay, so when an intergalactic cosmic ray goes flying through metal, it creates sort of gamma rays that come flying off it, come off these like stochastic spirals, and that causes like secondary damage. Okay, cool. So let's go have a look at that one. There was it. There it is. High dose. So I want to go in a bit more detail. So how many comparisons were they doing? Well, So, okay, we've got, so they were firing into these things. These, I'm not sure, I'd have to check whether this is the source of the radiation. I think it's the source particle they're using to generate it in the particle accelerator. So we've got 0.3 grays, 1.5 grays, we've got 3 grays. So you'll notice this is different to the centimorgans, I think it was on this one. But that's always the way. These bloody people, as you go into the radiation stuff, there's so many different ways of measuring radiation intensity, it's annoying as hell. But anyway, so again, we can do that classic thing where we can select the most variable, most significant genes, or the most variable, and go to significant. And so this is fished out from taken from all of these combined here. So it's a bit noisy, right? But it'll do. So these genes might be really interesting. They might be involved in the human response to radiation. Okay, cool. So what about in microgravity? So microgravity, you're sitting there, your bones aren't working, your muscles aren't working, your bones starting to waste, your muscles start to waste, muscles start to atrophy. It's a bit like lying in bed. And so now we're doing the overlap. We've got the genes we selected based on radiation. And here we can see 2 of those genes are involved in the human response to bed rest. So that could be a synergistic, that could lead to a synergistic effect. Microgravity plus intergalactic cosmic rays going through you could cause these loci to respond in a more extreme way, and thus is likely to have a greater effect on the biology. So these 2 candidate genes would be worth going into and trying to find out more information about in order to assess whether these should be monitored as when humans go out into outer space.
So, sorry to interrupt again. So like if you want to look at those 2 genes in a deeper way, is there like programs similar to like ePlant and databases where you can plug those 2 genes in and figure out their responses and look at their code and such?
Exactly that. Yep. So string, it'll work perfectly with. String is a good one to go to. I also think I had Shiny Go on my homepage.
Yeah,
didn't
you
load
did.
up just then, but that one too will take human gene identifiers. I think the other one I should point out is Human Mind. Human Mind? Is that what it's called? Yeah, it is. Because there's also one called Thailmine and Flymine and Wormmine. These websites are made by the University of Cambridge, England. Essentially, it's the same kind of function. You can put in a list of genes there and go from there. It will do some analysis and put out stuff that's related to them. Human Mind is good, but it's not as good as Thailmine. You know why?
Why?
Because Thailmine is about Rabbitops. But yeah, so that's a thing. That's there. Okay, so again, if you want to know more about them, okay, whoops, daisy, I just select those guys. I should say this one is still under active development. So if you do find anything a bit weird, do let me know. I think I've got most of it down. But I'm just adding more to it. So, but yeah, and then maybe we're going to go down. Just on the bottom here, we have these annotation sources. Ooh, that's the other one. Hang on. We could just go the top bit to human annotation data. So we've got those genes we selected from the bed rest study and whatever. This thing here is what I like to call a random prejudice generator. It's just a word cloud, but it's definitely a random prejudice generator. You can go through and get these summaries here, like as like in the pie chart format, trying to do your standard filtering thing, but that's just a waste of space. This thing here is again to get you all these summaries of these pieces of information, but it's on the other page, so again, waste of space. This thing here is the reason for coming here, which is, so I've got the human gene ID, which we selected from those studies. It's got the related gene name in mice, rats, worms, flies, and yeast, if there's an ortholog in yeast. So Maybe you don't want to deal with mice because you don't like cleaning up poo. So I think that's reasonable. Maybe you want to study this gene. Well, you could study its function in yeast really easily using flash lamps. Anyway, so close that, scroll down. And also there's these other little pointers here down the bottom here. So again, links to Expression Atlas. It's kind of embedded and it works, but I'll pop out, press, and open in a new tab. But this is a way of mining other microarray data sources. So for genes of interest. So this hasn't got the list of genes inside it, but you can search them in here. So maybe kind of makes sense. So these I'm just pulling in other tools and kind of hid them down the bottom here. There's it's a bit of a Easter egg. Benchmark tools are hidden in there nearby. So that's the gist of that one. So the moral of the story is you can see it's slightly different, but at the same time, you can see it's also really similar. And there'll be a new iteration coming out in about a couple of days. I've prepared another like 6 studies to go in. These Drosophila ones are now been fixed and functional. They're going to say work in progress as I wait for, and I've now fixed them. These are now operational, so I need to deploy the new database, but I wasn't ready. So the moral of the story is, if you do find things like this, do let me know. You can jot them down and send them to me an email, and very soon there'll be this one will be updated and this one will be fixed, and there will be new data, because this self-isolation thing has had an unexpected advantage. I've been able to catch up on some of my coding. Okay, cool. So as you can see, we've gone through TOAST, and we use TOAST to help explain the GeneLab Astrobiology Cross-Species Explorer. I've pointed out how the metadata is really important, and because there's so many studies, you've got to have a logical reason for looking at A and B, and you don't really want to read 200 papers, I guess. And the final thing that leaves is Mango. But at that point, I'm going to pause and go, any questions? No? You sure?
This would be a good time to ask some of those questions that you submitted ahead of class.
So current plans for growing plants on the lunar base. What lessons have we learned from experiments that have helped shape this plan? Well, the main thing is making sure the hardware is capable of controlling the oxygen availability and the water availability really closely. Actually, but not quite lunar base, but the next lunar lander, not the next one, but one of the new ones going up from JPL. The wizard Simon, he's advising JPL on what they're going to grow in there. So watch this space. First plants to be grown on the moon. Well, there's the ones that the Chinese did, but the video that they released was not actually the plants on the moon. The ones they released was the ground control. Not nice. Anyway, conspiracy. Okay, cool. We're about to become more data driven. How do scientists who lack these skills keep up with the changing world? You acquire new skills. That's what, so yeah, every single project I've gone through, I've had to learn different things. Start off as my PhD was like genetic engineering. The following one after that was like RNA-seq analysis, which was like a bit of like computer programming and bioinformatics. Then the one after that was a bit more different, like coding languages and lots of electrical engineering. I had to learn how to do wiring, and yeah, you just adapt. You just constantly learning new skills. And the longer you spend in the game, the more you realize you should make good notes early on. Yeah. Who assigns the functions of the genes in TOAST? Well, that's mainly TARE. So TARE is the Ground Ops Information Resource. For the last decade, they've been manually annotating a lot of the gene functions, pulling them in from other studies that have been done previously. So a lot of the assigned functions are based on previous studies. So the functions come from the biological research community and their databases. So Ensemble is probably like the main source. is the other major source. Ensemble is the European equivalent of TARE, but they go across all life kingdoms of life. I think I've shown this one. How it's possible to do cross-species analysis. The cross-species analysis, yeah, I demonstrated that one earlier. So just through selecting one dashboard, selecting the genes on that. At the moment in the cross-kingdom, you can't go between plants and animals and vice versa. I've got my own private version where I can go like do a pairwise comparisons, but it's much more difficult. So I haven't plugged that into the public version because it makes some weird glitch I haven't solved yet. And what is your approach to managing such large? Yeah, it's hard, and that's the metadata. I'm basically just going through the metadata lots to find the comparisons actually worth making, because there's so many comparisons. Like one of the GeneLab studies, they had 420 comparisons in it, just crazy. And it is incorrect. I spoke to the main author there, and that's not good. Oh, my internet connection has come unstable. Did you guys hear me all right? Am I talking to myself? The most important thing when doing bioinformatics is recording everything as you go. And this is actually the hardest part. So saving the files into the Excel file, into spreadsheets, saving the pictures into Google Slideshows, and then going in and writing text to describe it and what you're thinking. That final bit is the thing I fail at the most, to be honest. There's extra descriptive bits. I just kind of assume that I'm going to remember it, and most of the time I do when the pictures are there in front. But you really, it's annotating it as you go. So, because if I get a lot of data and you've got to put it into a slideshow to make a story, so it's turning the data into a story to make bits of it accessible. How do you collect newly input published data into TOAST and Mango? Is it continually updating? No, it's not. They're not continually updating. GeneLab is putting in the data. They process it. They put it online, and I have set up an API to identify it when they have new data that's up there. I put in the metadata, and that goes into the metadata apps. So the metadata apps will update automatically. But there are mistakes in the NASA GeneLab metadata. So I do like to go through every now and then and pull out their data, pull out the bits I'm interested in, i.e. the plants and the microbes, fix the mistakes that they have, tell them that they have these mistakes, and encourage them to fix it themselves in their metadata matrix. And then I put my updated fixed ones onto the Gilroy Life Science Lab website. Sounds a bit arrogant, doesn't it, saying that they have mistakes and I'm right and they're wrong, but I know that I'm right in the plant data, and I'm collaborating with Venkat from JPL to try and do the same with the microbial data. Got to have an expert in the field to spot the incontinuities. Usually it's simple things like someone having a capital S for spaceflight and another one having a lowercase s for spaceflight. It's there and it's annoying when trying to make filters to pass through the data. Where did the idea of the mass data of aggregation come from? A guy called Marshall Portersfield. Marshall Portersfield is the former director of the Life Science Space Research Program, SLIPS. He did a lot of early research on the space shuttle with Mary Musgrave into plant response to spaceflight. They looked at a few marker genes for spaceflight, like ADH, and they found some data which was quite compelling, but still anecdotal because they didn't have the big data that we now have at our disposal. So when he became the director of life sciences of NASA, he created NASA GeneLab with the goal of putting together all the life science data that had been scattered across all of the different locations in the cloud, with the question that if we centralize it, will a core pattern emerge? And it has, and that core pattern is changes to the atmospheric composition, i.e. more CO2 or less CO and less oxygen due to lack of mixing gases, is causing the mitochondria of all life in space to alter its function, you see gene expression patterns in relation to mitochondria changing in all things. And so it was Marshall Porterfield's idea to bring together the mass data, which has led to this pattern emerging. What's next? Could the system be tweaked for all topic areas? Could the system be tweaked? Yeah, actually, well, it doesn't have to. The system already exists for all project areas, and that is Array Express and Project Atlas. So you just go Array Express Atlas. Array Express Atlas, that's the term I'm after. So yes, the system already existed, and at the beginning of the program, when GeneLab was starting, I said, you're trying to reinvent the wheel. All you need to do is go to the expression array, and within it, bind together those studies in here that are already spaceflight related. And in the near future, my friends at Array Express are in the process of starting that now. And they're going to have a GeneLab button just down here. But the one thing about Array Express is it has got, say, 4,000 studies. It's really hard to collate the data, the metadata for 4,000 studies. And when you do it, say like GeneLab have done it for just, say, 200 studies or 250 studies, when you're an expert in the field, like, and you go in, you can still find mistakes that they've made. So in their 250 studies they've got, I can find in my apps mistakes and gaps really easily. And so when you have a larger amount of data, like the broader array, like they have, there's probably going to be some gaps in the metadata and some potential mistakes in how it was input. So the good thing about having a smaller list is that we can go in and we can curate the data and we can enhance that. And so that's kind of the best bit about the GeneLab program is that metadata curation that has occurred. But even with that, undefined there. So like assay technology. So like that's a really simple thing. Whether it's an Affymetrix microarray or an Alliant microarray or Illumina RNA-seq, you need to know that for every single study. And the fact that in this smaller GeneLab 100, 250 odd studies, 25 of them are undefined, that's bad. They need to fix that. Similarly, of those having undefined material type, i.e. of all 250 studies, 88 of them, they don't actually know which organ it came from, whether it's the leaf or the root or the leg or the mouth. Like, again, I'm in the process of helping GeneLab fix that. But the question was, could this be tweaked to go for all areas? And yes, it can and it should. The broader Array Express has that whole area like thing. But then we have to acknowledge there's going to be some gaps in this. And so I'm comparing and contrasting this stuff to GeneLab to try and find ways for GeneLab to grow and also to find ways to maximize the place where this has already occurred across all topic areas. I'm not sure. Does that answer your question, Sam? Not sure.
Yeah, it does. Sorry, it took me too long to unmute myself.
You know, I do that all the time. The amount of times I've been in telecons and I start talking to someone with a gap, and then I realize I haven't turned my thingy on. Oh, it happens all the time. So how do you see the TOAST and MANGO systems being implemented in the research community? So MANGO is currently, and TOAST primarily in use with Simon and with a couple of collaborators at the Kennedy Space Center. MANGO being used to provide easy summaries of the current microbial population. I'm just in the process of waiting to get my hands on the other really important microbiome data sets to plug into that. And that will definitely increase its value in the near future. And the goal is to basically provide people with easy access to the different types of microbes that are up there, so they can start to make informed decisions as to sort of experiments that we should do down here. Because looking at the genetic response to microbes versus like bacteria versus fungus, which one should you do, one or the other? And hopefully those clues will be found within TOAST and within Mango. And I know they are because I've seen them. And the problem is other people need to see them because there's so many open comparisons. We need lots of people doing it. Yeah, I see this as a jumping off point for future studies. I mean, it's a research platform in the bioinformatics perspective. It's a hypothesis generation machine. But ultimately, those hypotheses and the genetic patterns that come out of this need to be tested with future studies. That's why we created Flash Labs, those systems, and some of the cozy implements, the 3D clinostats that we showed you previously. Yeah, I really think Mango could be used in high school programs. I think like the interface of it is so easy to use. It's just the concept of a microbial like treasure hunt. Can you go in there and but then that then comes down to storytelling for high school programs, because it's easy to understand if you have a good story to tell. And searching for pathogens on the diagram table, I think, is an easy story to tell. Once you get down to the individual microbes, how can they start to give those microbes character? Well, it's the same thing with the genetics. When you first think of a gene, it's a weird ethereal thing of floating in the cloud, right? But once you've looked at ePlant, you can realize that genes have actual character. Like in a movie, you could give them character. And it's the same thing with microbes. And within Mango, there is the microbial directory, which allows you to give microbes character. And there's other resources online to do that, but I don't know what they are because I'm not a microbial person. So if you find good ones, do let me know so I can save the links next to Mango. I was an advisor on the pipeline for NASA GeneLab. So I was like a co-conspirator on that particular pipeline. I used to do the coding of the pipelines myself back in my last project, but now I've realized that it's more fun building the front end, like the visualization end, and just using other people's pipelines. The different types of pipelines, like statistical pipelines and analysis of RNA-seq, were continually evolving as I came into the field. So every year there was a new pipeline which claimed to have a better statistical like pattern. And so I've made hundreds of these bloody things. And now I've got bored of it. I just like using the currently believed best pipeline and just pushing that into my visualization systems. That's like a general computer programming type thing. When did you learn to code? Software carpentry and data carpentry. They're online, they're free. They have meetups in person, which are even better because I like to have a real person there. And so yeah, data carpentry. And so often the best thing to do is to just have a challenge to do with something with R. And that's actually why TOAST is so cool, because when you pull down the data, you pull down your list of genes, you have it there in an XLS format. Like you can read that into R, and then you can make a bunch of graphs with it. So yeah, it's just having something to do with it. Like if you really want to learn R or Python or any of those coding languages, it's having a real challenge that's worth getting your teeth stuck into to make it worthwhile for me. And that's the hardest part, because let's face it, most of the challenges they set you at school are boring. Who likes to be told what to do? I'd rather just investigate my own things and try and find a pattern which I think is awesome. And spaceflight is awesome. So finding genetic patterns in spaceflight data is awesome. So you can use this infrastructure built to pull down data, and then you can use some of the R coding infrastructure to plot these data. Kind of makes sense, right? On average, how long does data processing take after you get back? That's a hard one, and it depends on the experiment. How long does the data processing take? So pure bioinformatics, it can be relatively quick. It depends how good the data set is. It can take years. We had a data set and there were some weird anomalies in the data. One of the mutants was heterozygous instead of homozygous. It took years to find out, reanalyzing it again and again to find out which were the replicates that were incorrect and had a reason to discard. But eventually you get there. That's the beauty of the game. Persistence. Most important thing about being researcher is grit. Most people give up. That's all right. there's also a balance about knowing when it is the right time to give up. And no one can tell you when that is. With all things in life, you can keep hitting your head against that brick wall, and it ain't gonna budge sometimes. So that's why it is. I like to have lots of things, lots of options, lots of avenues, lots of tangents. Okay, who develops GeneLabs? Yep. So I think I described that. Me and my friends, we helped, like, there's a bunch of panels that had met up regularly over a space of like 3 years to discuss the current state of the art of omics-based technologies. And then there's a group of programmers at NASA Ames who then basically took the advice from this committee process to implement, and they are slowly pushing their way through the data. My friend Amanda, she is awesome, is one of the keys to this, as is Homer. He's another awesome dude who's also based in NASA Ames. They do the hard nuts and bolts. And then there's Sam. She's been in charge of the metadata curation and organization in general. Sylvain's the figurehead. It's mainly because he's got a great French accent, and and so when he tells you the things you don't want to hear, it still makes you feel so warm inside. No, he is awesome. He's a great radiation researcher, and radiation is the major showstopper in spaceflight exploration. It's the major unknown for going to Mars. So it's great having such a great gentleman in charge with such important knowledge in such an important field to help keep us going in the right direction, help us coordinate our sort of global effort. Yes. So I think that's the answer to all the questions, and that pointed out one last thing, which is just a quick go on. Man, I need to close all this shit down. See if this happens all the bloody time. It's like, ooh, that's an interesting network. I can't leave that. Ooh, that's an interesting network. I can't close it down. Close it down. Okay, so I have to keep myself in check sometimes. Okay. So what I want to just quickly last do is go to Mango. Mango's fun. Launch in a new tab. Again, classic thing. If you forget how to use it, you can watch this video. This one I think does have like nasty electro music playing at the top. Do listen to it, or turn off the volume, because I'm no musician. It's meant to be a placeholder to edit a voiceover, but I'll never do a voiceover, will I? So again, similar structure, a selection of dashboards to choose from, but we're not looking at genetics this time. Instead of having lists of genes, we're looking at the microbiome, so you have lists of microbes. Simple. Job done. Simple, same kind of thing. We have different locations on the station, so we can look at data based on location, and it kind of makes sense. Some people like the phylogenetic tree, and so maybe you think of things as species, or genuses, phylums, domains, all that kind of stuff. This is for your classic microbiome person. I ignore all of that. Quite like the species. To see that kind of thing. So phylogenetic tree kind of laying out, you got your cellular organisms, you've got your bacteria, your archaea, your eukaryotes like your funguses, that kind of stuff. You got your non-cellular organisms like your viruses and all those kinds of things. Oh yeah, if you want, you could fish out all the viruses on the space station. That would be fun. Yeah, we'll do that. Okay. Okay, so we'll come back to that. Viruses are the game for today. So you can look at the data in a sort of filtered centric fashion as well. So if we're going to choose a location, we've got to get familiar with a location. So first of all, we're going to fly around the space station, because why not? Oh, I've always wanted to go to space. What's down there? Where are we? Oh, there's no 2. Destiny module. Oh, I love Destiny. Do you know why? Why? Because there's a computer here. No, it's not. Because this is where the brick, our brick 18 was resided when we first, brick 19 when we first did the study up. But today, we're going to focus on this guy over here, the dining room table. You can see that there's a ketchup thing on the surface. So we're here in node 1. Node 1, dining room table. Okay, so we're going to think about the node 1 dining room table. Node 3, resistance device. No, we don't need the exercise device. We're going to go to dining room table. So we've got a few different dates to choose from. And this PMA treated and PMA not treated. We'll have a look at those in a moment. So not treated. Dun, 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 dun. Okay, so these are the microbes on the genome table on the 4th of July or 4th of March on this side. And then on this side here, this is the quantification of those same microbes on their advanced resistance exercise device on that same day. Ooh, kind of cool. That's a plant pathogen. That's a plant pathogen. Okay, cool. That's kind of neat. So let's have a closer look at the dining room table. I'm gonna use my plot, aren't I? I do this all the time. Resistance, exercise, dining room table, node 1. There it is. There's the photo. Jolly good. So we're at the right place. We have our 3 time points to choose from, like a transect through time, because it's sampled on 3 different occasions. Then on the bottom here, so this is that quantification coming up. We have the different time points. Non-PMA treated and PMA treated. So if you treat the microbes with samples with this PMA chemical, it will destroy anything that isn't living, essentially. If the membrane is not intact, it'll get destroyed. So this here was selecting for living samples. Okay, but whoa, there's loads of them, man. Okay, what am I gonna do? How am I gonna select that? Okay, I'm gonna go to, let's have another look. Let's think about this. How many species have we got here, potentially? Holy guacamole, got 25,000 species are detected up there. That's a lot. We don't want to look at the exercise resistance. We want to look at the dining room table on the 4th. Should we do the ones that are alive? Let's use PMA treated. Oh, look, we're down the bottom here. You can see that. There they are. I move up and down a little bit. Oh, there. Okay, so because of the way it's done this weird thing, I'm just going to click on that. Oh, no, it's not done right. Can't. I'm going to do that. So there we go. So now we've selected all the ones that are expressed a little bit at that location that are found there. Not expressed, sorry. That's me thinking about genes again. Sorry. Once a geneticist, always a geneticist-ish. So now I'm just going to go down a few layers. So the top 24. Again, we've got a pairwise comparison here. So we're still looking at the exercise resistant device there. So I'm going to go dining room table. And then I go down, so not PMA treated, and now this is the DNA PMA treated. So these are alive, these are dead. Okay, cool. So there's correlation between those that are dead and those that are alive. That's interesting. I didn't realize that, but that's good. We can see what the main players are. Okay, so I am not hugely familiar with microbes. I don't really know what any of these things mean. I can't put it in context because I'm not a microbiologist. So do you know what I'm going to do? I'm going to go to a database. Plant pathogen interaction, microbial directory. So fortunately, none of them are on here. This is good. Okay, I'm just going to turn that field off for a second. So the plant pathogen strain thingy, hang on a second. So this is a count of all of the pathogen strains that have been detected on the space station. And then down here, this is the prevalence of disease-causing microorganisms found on the space station. And this down here is the microbe directory. So all the different all the different pathogens and such, and the host species are all kind of listed here. Okay, so we can use this as like a filter. And so those filters along here. In addition, plugged into the database, we have the pathogen host interaction database. So we can take a bunch of things like this triticum, so it's like a wheat variety. And you can see all those like other terms down here. These are all the microbes that are pathogens for it. So we can start to find the pathogens that are in orbit and their host plants. And that information's here, and again, it's all can be downloaded. Okay, cool. So So what I'm going to do is I don't care about these ones because I only care about plant pathogens. Now it's over here, plant pathogen. Okay, cool. So I'm now pre-filtering for microbes that are described as being plant pathogens. And then I'm going to go back and look at the species tab. Do I still have those locations loaded up? Pretty much. So, so from those species that we were selecting earlier, just a few of them, because we were selecting for what was on the dining room table, right? But from within that selection, 1, 2, 3, 4, 5 of them are plant pathogens. So if I was advising farmers on the space station, I might want to tell them about some of these plant pathogens. Kind of makes sense because if the astronauts are going to the dining room table to have their dinner, they're likely to come in contact with these, and they're then likely to float over to their plant or dinner grow area and might take it with them, right? Again, we can pull down the data. We have the quantification of these microbes in all of these different locations. And again, we're basically just using these systems here to filter it down to a small enough number where we can start to go, why is Methylobacter mesophilicillin on the dining room table? How might that affect plants in the dining room table? And the plants in the in a grow area. This is like another way to view these data. I'm gonna have to upgrade these shortly. Old plots. They don't like the code doesn't like the names at the bottom here if you have too many. Anyhow, so yeah. Whoops, I shouldn't have popped that open. Sometimes that happens when it takes a while for something because there's a big amount of information there, and I'm so impatient. I've already lost my train of thought. I forget why I popped it open. I should probably just leave it. So yeah, sometimes you have to be patient with the software. So what's next? So as you can see, we went through, we filtered down, we found some. Oh, come on, killed it. That doesn't normally happen. Well, that sucks. It's because I can remember I said like when you have doing a live demo, it's when you're most likely to find a glitch. Anyway, so I'm just going to refresh that because I'm that impatient. Anyway, so the moral of the story is you can use Mango to go in, find some microbes, pull them down, see where they are on the station, give them character. But then, well, my whole computer's glitched up. It's because I've got Mango open twice. I've got it open on this thing and on this thing. Refresh. Okay, there we go. It's loading up now. Yeah, try not to have anything open twice because I had it open on this web page. It was loading in here, and then I had it open on the other page, so it was loaded up twice, and I think that's why it froze. Yeah. Okay, last final little details to add. I do have some genetic data in here. I've just started putting in the transcriptional data in the same way that you saw in those other studies. So this is Bacillus subtilis, transcriptomics. There should be a link to the paper, which I haven't put it in there yet. I should fix that. And the thing I wanted to look at was viruses quickly. I forgot that. That's the other thing. Virus. There's not many viruses up there that were detected. But one of them, this BAC gamma retrovirus, is actually kind of similar to corona. Oh well, don't tell anyone. And it was found in the ISS air filters, but it's not on the surfaces. But that's kind of cool. So that's topical. Any questions? because that's the gist of that, really. Things to point out. I'd recommend, if you get a chance, watching this. So this is Darwin Newman. She's the former director of NASA. She was awesome. This was one of the. This is just like a 30-minute or so seminar that she gave at the American Society of Gravitational Space Research in Washington, D.C. A couple of years ago. She's awesome, and it's really, really great stuff in there. And this dude is completely bonkers. This is some crazy, unheard of, like, space nerd who's just made a really awesome video, which I think was worth embedding. You don't have to be the director to say, like, awesome things. So I've just kind of hidden loads of gems in this on the website. In the near future, there'll be a Mango microbial metadata app just there next to it. And in the future, Mango will grow. And that's why I need to call Nitin back to work out what's next. Any questions?
Can I go over their assignment with them really quick?
Yep, indeed.
Okay. Maybe if you have other ideas, you can add to it. I'm going to share my screen then.
Sure thing, I'll stop my share. Because that's about it, really. I think, in a nutshell.
Yeah. Can you guys see this Word document? Okay. So I thought that for this part of the class, which will be sort of more laboratory focused, you know, initially we were going to be able to do our gravitropic assays in the flash labs, but now, since those are stuck at Carthage, we'll just basically be developing an experiment and writing a proposal. So what I'd like you to do is to make 10 PowerPoint or Google slides that describe a candidate gene that you've identified in a Arabidopsis for a potential gene to mutate in a Arabidopsis, and then study the effect of that mutation in either a gravitropic or a thigmotropic, a light response assay. So the idea behind it being Google Slides is after I get a chance to review them, I might then have us probably either next week, like a week from today, or after Easter break, have you present on the genes you've identified. So I'd like you to go through TOAST the way that Dr. Barger showed us today, and the instructions that I provide in the lab folder. Try to narrow it down to one gene you're interested in studying. And in your slides, make sure you describe the filters that you use to find that gene. You know, if it was from certain experiments that were done on the space station or shuttle missions. Try to say like what experiments you compared. If you did like dark versus light or different upregulated or downregulated genes, just sort of walk us through that process in your slides. And then he was showing us how to download different images from TOAST and some of the other apps. So you can click on those green arrows if there are neural networks you want to show, or Venn diagrams, bar charts, or the really nice figures in ePlant showing where the genes are expressed in different tissues. Since we are looking at either gravitropic or light responses, you might want to sort of focus on genes in the roots, especially for the gravitropic response, since those are where typically like we would have measured the root turning if we were still doing those experiments. State what your proposed experiment would be. So if you were going to use the flash labs, would you have changed the color of light, the amount of light, like the timing, or would you have done the gravitropic assay? And then try to provide, you know, some of the potential significance that you could gain from this study. Like what would this help you understand in terms of the Arabidopsis response to spaceflight? And then lastly, just provide some references in the slides. So you guys have any questions right now about that?
I have a quick question. So let's say we do like our filtering process and we find a gene for that's a good gravitropic candidate. And then we research it further, like on PubMed or something, we find that people have done wet lab results on it and figured out, oh, it responds to this in a gravitropic assay. Is it okay if we like include that, or should we find something that no one knows anything about yet?
No, I think you can include that. It would be good to validate, and I'm sure like your settings could be different. Maybe you change the light or the timing of the experiment.
Sure.
Richard, do you know, like, I don't know what the chances are that they would find a gene that's already been validated in just the gravitropic assay.
It would be awesome if that happened, to be honest.
Yeah.
Yeah. I mean, it's plausible, but I don't think I've ever been able to do that.
Okay.
I mean, yeah.
Yeah.
So, but it is possible. There's so many potential comparisons within it. I'm sure there's one of them which has a logical reason as to why there's like specific gravity response genes. Yeah, GLDS 251, I think it is. They grow the Arabidopsis plants in a centrifuge on orbit, so it has 1G, and then they switch off the centrifuge and they wait half an hour for like the gravity to kind of for it to slow down. So there's this fractional G-forces that slows down to microgravity, and then they switch on a blue light, and the root goes away from the blue light, and they harvest the root tip. That data is in the in TOAST. So, and it's in Cross Kingdom too, I think. If not, it'll be the next release, which will be out in a couple of days. And so you would think that particular data set might be one where there would be loci that would are likely to be involved in a gravitropic assay on Earth, right? Because it's switching off from 1G to micro G is a stimulus akin, which is more similar to a 9-degree orientation, right? The other ones, when they've been growing up there for a long period of time in complete microgravity, like that's a drowning, suffocation kind of thing. But when you've been growing in a centrifuge, spinning with artificial 1G, those are the studies that are likely to have the genetic pathways that link to gravity perception in a way which can be detected in gravitropic assays and follow-up. So the clue there is the European Modular Cultivation Unit, or EMCS system, European Modular Cultivation System. But again, that's the kind of thing that those kind of clues are the ones that are in the metadata. I just memorized the metadata, so I thought I'd give you a clue that's in there. Oh man, I must be slightly autistic. Moving swiftly on.
I'll show you guys as well. Are you seeing my web browser? Yeah, so for next week, Tuesday, we're going to read an article on bone physiology using zebrafish as a model for bone density loss, sort of like osteoporosis. So that article is posted, as well as the assignment, just a couple questions to answer. But you'll want to work on that, and then trying to identify the candidate genes. Again, if you go back here, under the lab folder, link number 7, I have the previous kind of protocol that walks you through some of the steps that Richard showed us in TOAST today. But I'll also upload this video from today. If you want to go back and see what he showed us, you can get to the relevant part for the different tools you might be interested in using. And then lastly, I am going to post, probably tomorrow, I'll get it published for you on Schoology. I'm going to post another little lab exercise I want you to do for next week, starting to analyze some of the sample data we have from Flash Labs. So you're going to measure like the angle that the root changes in response to a gravitational direction. We're going to manually do that using Fiji. So I will post that lab and the sample data set. There's 2 sample data sets in this folder. I'm just going to have you start with wild type for Tuesday, and then we'll come to class Tuesday, discuss those, identify maybe any issues you had.
Quick question.
Yeah, go ahead.
If any of you like Python, like computer programming, the software So the way that we're going to analyze them in ImageJ is the best way to do it from learning how biology and plant science is usually done. But there's a new opportunity. If any of you know Python and would like to practice, there's a new piece of software called RootNav2 that in theory you should be able to upload those images to and run it through that Python script. And it has a machine learning algorithm that's been trained to automatically detect the plants and quantify them. It's something I'm in the process of trying to get installed into Cyverse in the cloud to make it accessible. But yeah, if any of you guys like Python or want an opportunity to practice it, those images there provide an awesome opportunity for you to do that. Yeah, over.
Okay, good. And then there was a software called Root Trace a few years ago. Is that still working, Richard?
That is, yeah. That one is on my GitHub account. There's a version you can download from there really easily, but it has to run on a Windows operating system.
Okay.
In the same way that next to that, on my GitHub account, there is a RootNav software, RootNav #1. So that's what it was the predecessor to RootNav2, and it has a really nice graphical user interface for measuring like the root system architecture and providing information like the root tip angle. So ImageJ is what all like biologists use. It's brilliant. Whereas RootNav and RootTrace were both designed specifically for Arabidopsis, and so they have use extra little bits in them. So if people want to use those, they have to have a Windows operating system, and again, I can help navigate your way through those if needs be.
Okay. Yeah, I thought we'd start with the Fiji or ImageJ kind of manual analysis first, and then if we want to get more into image sets, we can move over to Root Trace. I don't know if everyone has a Windows machine though at home. That might be the issue. I don't. I only have a Mac, so.
And that's why I'm really excited by this RootNav2 because it runs through Python, so it can run on any machine. And in the near future, again, I'm trying to get it up in the cloud. It's in Cyverse, so it's fully automated. But yeah, in the meantime, if anyone knows Python and likes to do Python coding, feel free to get in touch about that. And yes, we can overcome that challenge together.
Okay. All right, everyone. So I'll post the research proposal instructions in the lab folder, and then tomorrow I'll publish the sample data set and the directions for using ImageJ or Fiji, so you can play around with measuring the root tip angle. I'll send out an email as well with further directions on that. Does anyone have any questions right now? Okay, the bone article is published already, so if you want to start reading ahead for next Tuesday, you can do that, as well as complete those written questions.
Sam has a question.
I have a question unrelated to some of that. I was just wanting to ask about the articles that we submitted for today.
Oh yeah, I didn't get to look at everyone's yet. It's been kind of hectic around here, but I'm hoping to look at them. Is that your question, or?
I was just kind of hoping, like, so when we submitted our articles, like, could we like start now on working with that, or should we wait for approval?
If you had submitted multiple articles, why don't you wait for my approval? If you submitted just one, and you're pretty confident you want to go ahead with that one, you can. I'll probably have time tomorrow to look at them. So if everybody wants to wait until tomorrow evening to hear from me, I can do that.
I think as of this morning, I was the only one who had multiple articles.
Okay. I know Melanie had emailed me too as well. So, but Sam, you just had one. What? Yeah.
I think so. I had one, but I had a backup just in case it didn't work.
Why doesn't everyone just wait till tomorrow evening and I'll verify that your articles will be appropriate and that we don't have too much overlap on one topic? So, yeah, I'll send out a follow-up email tomorrow then. Okay, thank you.